This is the QX700. It's the next leap forward in digital PCR instrumentation, and the first device that works with laboratories, not against them. Let's take a look. The QX700 is a fully autonomous, all-in-one, highly compact system. You can start experiments wherever, whenever, and you can make them as simple or as complex as your application requires. As a routine user, it takes about 5 taps or clicks to get going. But as an expert, you have as many as you like. A single cartridge plate holds up to 3 cartridges, and 1 cartridge holds 16 samples. The QX700 comes in 3 configurations. The QX700E for essential digital PCR, allowing you to load 1 plate with up to 48 samples per run. The QX700S, capable of loading 4 plates simultaneously with random access, which results in 192 samples per run. And the QX700HT for high-throughput workflows, enabling random access loading of 8 plates concurrently. Up to 384 samples are processed automatically by the HT's dual thermocyclers. So you can choose to work on your own, work with a colleague, or bring in the entire lab, all on a single instrument. If you insert your last plate as you pack up and let the HT finalize it overnight, you could process up to 768 samples per 8 hours of lab work. Let's talk colors. The QX700 is the first digital PCR instrument to support 7 colors, with a brand new purple as an extra color. And it doesn't stop there. Using higher-order multiplexing, you will be able to combine colors to detect well over 20 targets in a single chamber in the near future. And all this builds on our signature chip, the RDG16 cartridge. It's our most flexible and accessible digital PCR cartridge yet. It's much more sensitive than qPCR out of the box, and it's built to work exactly like regular plates. And when the QX700 is done, it immediately runs automated QC checks right on the instrument, giving the control you need exactly where you need it. And as with all of our software, all acquired data is available to you, from the raw images of our droplets down to the automatically post-processed digital PCR dot plots. Once you're done, simply validate the data and continue to analysis software. On analysis software, you can view data across the dimensions you need with a 1D, 2D, and 3D plot. You can control populations with automated thresholding, manual thresholding, or draw up your own polygons to create custom populations. And when you're ready, you can export to whatever file format you need and move to the next experiment. So that's the QX700, the first digital PCR instrument of its kind, bringing powerful digital PCR to everyone in a workflow that's a breeze. For those of you running high-complexity experiments, for those running hundreds of samples a day, for those of you who are just getting started, and for those that are anywhere in between.